and we're going to be talking all about biotechnology in this section. And in this first lecture, I'm just going to start by giving you an introduction of what is biotechnology and just set you up for some of the things we're going to be talking about in later lectures. So what is biotechnology? Biotechnology can be thought of as utilizing biology for human purposes or for our own human needs. And certainly there are things that you have heard about. One that I should have included on this list would be like CRISPR, which is a technology where we can actually take and edit genomes of whatever organism we want. Basically, we can change whatever genes we want fairly easily and fairly cheaply. In fact, there's news coming out lately that people in China have been born after being genetically modified with CRISPR, which poses some really severe moral issues. But there's also DNA used in the courtroom where people who have committed crimes are either exonerated or convicted. And frankly, this is going to become, I think, very much more common in the near future, whereby if you're going to commit a crime, you're probably going to be caught with DNA. There's also sequencing of the human genome that you can do. So you could go home and you could get your genome sequenced for about $100, and they can tell you lots of information. They can say, hey, here's what color your hair probably is. Here's where your family is from. Here's a bunch of different genetic conditions that you might have or things that you might want to look out for. And you have the option when you do those things you can do stuff like just get the ancestry. So you don't have to worry about the ramifications of having potentially some genetic condition if you don't want to. But personalized genomics is really cheap, and it's really interesting. We've also got treatments for different things like dwarfism, cystic fibrosis, muscular dystrophy. Many others belong on this list as well. There are genetically engineered food or drugs that you eat every day. I can pretty much promise you that. If you've eaten corn in the last, actually, for most of you guys, if you've eaten corn in your lifetime, you more than likely have eaten genetically modified foods. If you've eaten soy, you've eaten genetically modified foods. If you've drank an energy drink, you've probably drank the result of some genetic modifications. But as I was saying before, with things like CRISPR, there are always hazards and there are always potential downfalls to these things. And I think it's really important for scientists to be very careful and be very cautious with the things that they begin implementing, because these things can have huge impacts and unforeseen consequences that arrogant people will never be able to see. And so it's kind of up for debate if you think that government regulation is what's required, or some folks might think that it should just be up to the scientists themselves. I have opinions on it, but I don't really know the right answer. So here are a few examples of biotechnology today. Over here on the right, you have salmon that are able to be grown up much faster than they would in nature, which is a pretty cool thing. We have these goats that have been genetically modified to produce different kinds of milk, or milk with different nutrients in them. And so like this one here it's got milk that has human proteins that prevent blood clots, which is really fantastic. Here's a cloned horse up here. So this clone is an exact genetic copy of its mother. And this is kind of a fun one. They took a gene from a firefly that bioluminesced. They took this gene and they put it into a tobacco plant. And so now you can see the tobacco plant glows in the dark, which is really cool, kind of fantastic in my opinion. So biotechnology is very often the use of living organisms to create products or facilitate processes. And so an example of this is we often use biotechnology to create human insulin, because the way we used to do that was we would go and get a pig and we would find its pancreas, which is going to have a high concentration of insulin, and we would grind that up and we would try to use that for people who were diabetic. Unfortunately, it was not a perfect match. It wasn't a perfect way of doing it because it's pig insulin and not human insulin. But now what we've done is we have taken the human insulin gene and we've placed it inside of a bacteria. I'm going to talk about this more later. But we've taken it, we put it inside a bacteria, and now we grow human insulin and other products with bacteria. There's also this thing called PCR, which I talked a little bit in class. I talked about the guy who invented it and how he's a little bit unique, we'll say. And you can use PCR to make copies of segments of DNA. And without PCR, you would be limited to only doing one test with whatever piece of DNA you had. So if you had blood, you would only be able to do one test. But now that we have PCR, we can make copies of the DNA that we want to make copies of, and we can do as many tests as we want, and it's fairly cheap and it's fairly quick. So in the next lecture, we're going to start diving into these different aspects. There are 4 different basic kinds of biotechnology that we're talking about. You have transgenic biotechnology, which is taking DNA from one species and putting it to another, which is like what I talked about a second ago, where you took human insulin genes and put them into bacteria. You have reproductive cloning, which is production of animals using adult cells. So again, like the horse I showed earlier, we took some DNA from a random horse cell. I don't know where exactly, but it could have been like the horse's ear, put it into an egg, and got an exact genetic copy. We have forensic biotechnology, which is what's often used with that idea of PCR, whereby you can figure out who the culprit is, or you can figure out who the culprit isn't and maybe exonerate someone. We also have personalized medicine, which is medical treatments tailored to an individual's unique genetic makeup, and you can figure that out using biotechnology, and you can treat them better using some of these biotechnology ideas. So in the next lecture, we're going to start getting into transgenic biotechnology.